Hi to all of you, welcome back to the sixth lecture of the biosignaling series of lectures. In this lecture, we'll be discussing hormones, particularly focusing on the cGMP and calcium signaling in this lecture. So, brief introduction about the cGMP. Cyclic GMP is similarly a second messenger as was the cyclic AMP, and it's used in a diverse cellular growth, contractility, inflammation, sensory transduction, as well as neuronal plasticity and learning. And some of the drugs like nitrous oxide releasing organic nitrates, which lead to the angina pectoris, also utilize this signaling in mediating their effects. And specifically some drugs which are called as the sildenafil, which is used for the erectile dysfunction treatment, serving as the inhibitor of the phosphodiesterase 5, also mediates its effect through cGMP signaling system. Like cyclic AMP molecule, cyclic GMP molecule is also produced by cyclases, and here the effector enzyme which is responsible for the generation of cyclic GMP from the GTP moiety is guanylyl cyclase. Guanylyl cyclase is in response to the specific kind of signal molecules like nitric oxide, guanylylin, or atrial natriuretic factors. When the receptor gets activated by these signals or by these ligands, guanylyl cyclase gets activated, which in turn produces the cGMP as the second messenger for the response to these hormones. The basic difference between the guanylyl cyclase structure and the adenylyl cyclase structure is in its simplicity and as presence in two different types in the effector or target cells. It can be as soluble receptor or as a membrane bound receptor. Soluble receptor is heterodimeric, as you can see on the left hand side of the picture, and the membrane bound is usually functional when it is present as a dimeric form called as a homodimeric structure. But when hormone and when the ligands are not bound to this kind of an guanylyl cyclase membrane bound structure, it is present as a transmembrane single spanning protein which contains three different components. The N-terminal part, which is located outside the cell in response to the environment, is called as ligand binding site, ligand binding domain or exterior domain. Then there is a transmembrane domain, TM domain, which is spanning the plasma membrane. Then the third one is the C-terminal intracellular domain, which relays the activity to the bound GTP proteins downstream. So this is the basic difference between the guanylyl cyclase and the adenylyl cyclase. Further, the soluble form of the this receptor is coded by two genes, while as the transmembrane membrane bound guanylyl cyclase is synthesized by 7 different genes in humans. cGMP signaling is actually based upon the type of a target cell, which is in response to the different kinds of the drugs or different kinds of ligands or hormones. In kidneys, what happens is that the cGMP molecule are produced in response to the atrial natriuretic factors, which are produced by the heart muscles in response to the increased blood volume. When the volume of a blood increases, heart muscles respond to this and they secrete more and more of atrial natriuretic factors. These atrial natriuretic factors then get to the collecting tubules and collecting ducts of the kidneys, where they activate the guanylyl cyclase located on the surface of these cells. And when this happens, what happens in response is that the cGMP is produced, and cGMP then triggers the increase in renal excretion of the sodium ion, and consequently in response to this, water is consequently excreted more. This results in the decreasing in the volume of the blood finally. So in kidneys, the response is dilation of the afferent arterioles, and then there is also a suppression of the reabsorption of the sodium, so that more and more excretion of sodium will happen. Talking about the endothelial cells, which is the curious case, is that this cGMP signaling is in response to a gaseous second messenger or gaseous hormone or ligand, which is actually the nitric oxide. Nitric oxide is produced by a specific enzyme which is membrane bound, that's called as nitric oxide synthase. What happens actually is within these endothelial cells, in response to the nitric oxide, the cGMP phosphorylates certain proteins and reduces the endothelial permeability for fluids and macromolecules. The actual relay of the signaling is via an hormone which is acetylcholine, which actually binds to the external surface of the endothelial cells, results in the transient increase in the calcium concentration within the cell, which in turn will activate the NO synthase, and then nitric oxide synthase in turn produces more and more of nitric oxide from the arginine. This nitric oxide then binds to the soluble guanylyl cyclase and thereby activates it, producing and converting more and more GTP into cGMP. Then cGMP does its function by activating the protein kinase G type, and this protein kinase G type results in the change in the cytoskeletal structure, resulting in the change in the permeability of these endothelial cells. And the phosphorylation by protein kinase G happens like similar way as for the protein kinase A effectors, resulting in the phosphorylation of serine and threonine residue of the proteins which are downwards under the influence of these guanylyl cyclase as well as protein kinase G. There are wide varieties of proteins which are under the influence of these protein kinase G, which do a wide variety of functions. The third kind of a signaling which is seen in the human body is in intestinal cells, which utilizes cyclic GMP as the second messenger, and this is in response to the guanylin. And this guanylin actually activates the plasma membrane bound guanylyl cyclase to produce the cGMP molecule, and this is actually a regulator of the chloride secretion in the intestines. What happens in the intestine is that this GC receptor is activated and is a non-conventional target for the heat stable endotoxin of E. coli as well. This endotoxin produced by the E. coli causes a drastic elevation of the cGMP whenever there is an pathogenic infection. This results in the higher levels of cGMP than the normal, and the net effect will be to mediate and affect the increased chloride secretion, and consequently there will be a decreased absorption of water. Both of these will lead to diarrhea in the intestinal cells. So the E. coli toxin affects the humans by regulating the cGMP levels and hence causing more and more increased secretion of chloride in the lumen of the intestines, resulting in the diarrhea. As happens in the cyclic AMP signaling, where there was a protein kinase A involved in the relay, here protein kinase G is involved for the relay of the information downwards by the action of a cGMP, and it almost works in a similar manner as the cAMP molecule. It binds to a single polypeptide of both catalytic and regulatory domain of this enzyme, which contains the single monomer unit, in contrary to the protein kinase A, which was a tetrameric unit. Here the protein kinase G is a single polypeptide containing both catalytic and regulatory domains, and when it binds to the single molecule of a GMP, it results in the change of conformation, which in turn will expose the catalytic domain and bring out the phosphorylation of the serine and threonine of the proteins which are downwards under the influence of protein kinase G. These usually are the metabolic enzymes which are regulated by phosphorylation and unphosphorylation, and most of the metabolisms which are under the influence of protein kinase G are regulated like this. So as you can see in the current panel, the cGMP signaling is by response of guanylyl cyclase to the effector ligands, either guanylines or nitric peptides, which in turn will activate the guanylyl cyclase enzyme, causes conversion of more and more GTPs to cyclic GMPs, which in turn will cause the activation of the protein kinase G. Protein kinase G results in the phosphorylation of number of different kinds of metabolic proteins as well as cytoskeletal structures to do the function. Now we will be focusing on the calcium signaling. Calcium signaling is important. Why? Because this is the signaling which actually not only stands alone but also crosstalks with other signaling molecules as well, other signaling second messengers as well. Under the normal conditions, what happens is that there is a specific steep gradient between the two membranes of the cell. First membrane, which is the general membrane, which is called as plasma membrane, and second membrane is the organelle membrane versus cytoplasm. Under normal basal conditions, what happens, the extracellular calcium concentration is around 5 millimolars per liter, whereas the intracellular concentration is maintained at 10 times lower than this, 100 times lower than this, that is 0.05 to 10 micromoles per liter. So the gradient is usually 100 to 1000 times, usually if you see. And what happens also is not only the extracellular calcium concentration is high, but also the organelle, which is mitochondria and endoplasmic reticulum, also serve as the storehouses of the calcium, particularly the endoplasmic reticulum. So there are two gradients which are maintained within the cell. One gradient is between the cytosol and the external environment. Another gradient is between the cytosol and the endoplasmic reticulum cytomatrix. So in spite of this large concentration gradient, a favorable transmembrane electric protection and gradient is also maintained, so as the cell will do its normal functioning, and the calcium is restricted from entering the cell either from the outside or from the endoplasmic reticulum. So how does a cell regulate these cytosolic concentrations of the calcium at a very low level? Is by utilizing the four calcium channels mainly, which help in the regulation of the calcium levels in the cytosol. Two are functional in basal conditions. One is the sodium calcium channel, which is present in the plasma membrane, which transports the calcium out of the cell from cytosol to the external environment. Another is the CREB, which is present in the smooth endoplasmic reticulum membrane, which carries the cytosolic calcium into the endoplasmic reticulum, maintaining it at a very basal level at the normal conditions. But when the hormones are present or the neurotransmitters are present, they activate and utilize this gradient and open up to move the calcium either from outside to the cytosol or from the endoplasmic reticulum matrix into the cytosol. These are voltage dependent calcium ion channels like that of the present in the plasma membrane, or the ryanodine receptors which are present on the surface of the smooth endoplasmic reticulum, which are responsive to certain second messengers like that of IP3, which I just covered in the previous lecture as well. So what happens in the calcium signaling is that there are three ways by which we can change the cytosolic calcium concentration. First and foremost is by activating the receptors which are present in the plasma membrane. These are specific ligand gated channels which are responsive to the hormone signaling. When hormone binds to this, these channels are opened up. They help in disrupting the calcium and causing a transient spike in the calcium concentration in the cytosol by moving the calcium out from the outside into the cytosol. Second is by indirectly promoting the calcium influx by modulating the membrane potential of the plasma membrane, which in turn will cause the voltage gated channels to open up, which we just covered in the previous slide. So by these two things, either by direct activation by the hormone or by indirectly changing the voltage of the plasma membrane, both will function in moving the outside extracellular outside calcium into the cytosol and causes transient increase in the calcium concentration. And secondly is by activating also the ryanodine receptors in the endoplasmic reticulum. This is done by IP3 particularly. IP3 is a second messenger of the phospholipase signaling as well. So IP3 will move, bind to the these ryanodine receptors to the endoplasmic reticulum and cause the movement of calcium, stored form of calcium, back into the cytosol, resulting in the transient increase. And hence when calcium is spiked and calcium concentration is increased, it in turn will activate the specific kind of kinases which are called as CAM kinases, calmodulin kinases or calmodulin calcium kinases, which in turn will do their function of phosphorylation of the downstream proteins. So further down, the calcium signaling is actually mediated through their workhorse, which is calmodulin. It functions in a similar way as that of protein kinase A or protein kinase G. It is a calcium dependent regulatory protein. It is 17 kilodaltons in its molecular weight and is homologous to muscle protein troponin C in structure as well as function. It functions only when it is bound to calcium. It requires for its whole expressivity and for its whole occupancy of 4 calcium to its binding sites, which in turn will cause a marked conformational change leading to its activation. When it's activated, it is an enzyme similar to that of an kinases, and it does the phosphorylation of a number of downstream proteins on their serine and threonine, thereby activating it. Particularly, it mediates its action through AMP kinase. AMP kinase is one of the important kinases which regulates the metabolism, energy homeostasis, gluconeogenesis, appetite control, and so on and so forth. It also mediates its action through CAM kinase 1 or CAM kinase 4. But major metabolic function by calmodulin is mediated through AMP kinase. If it is a nutrient response like protein translation or bone mass accrual or something which is related to the neurological function, then it is mediated through CAM kinase 4. So kinases are important under the influence of calcium. So calcium mediates its action through its workhorse, which is calmodulin. Calmodulin particularly is involved in regulating various kinases as well as enzymatic ones which are responsible for cyclic nucleotide generation and degradation. In addition, this calmodulin calcium complex also helps in the structural element regulation and their function. For example, actin myosin complex requires for smooth activity of contraction calcium and calmodulin. Microfilament mediated processes are also regulated by it. Mitosis, endocytosis, all these functions which require certain cytoskeletal structures require calcium for their function. And via its AMP kinase pathway, it mediates the regulation of number of kinds of metabolic enzymes like the enzymes glycogen synthase pyruvate kinase, pyruvate carboxylase, pyruvate dehydrogenase. All these are mediated and regulated in their function whether to activate or not to activate by AMP kinase. This is the metabolic arm of the calmodulin. So when metabolic arm of the calmodulin functions, it functions through AMP kinase. As I already told you that calcium signaling is actually under the influence of certain other signals, and particularly the calcium signaling is a sub-arm of phospholipase signaling. And we know from the previous lecture that the phospholipase signaling is activated by binding of an hormone or ligand to a specific GPCRs located on the surface of the cell, which in turn will activate the GQ type of the G protein. And this GQ type of the G protein will then activate the membrane route phospholipase C beta 1. This will activate the phospholipase C beta 1, which will in turn cause the production of a second messengers IP3 and DAG, and they will activate protein kinase C. Also, protein kinase C will then do the activity of their own mediation through kinase activity. However, IP3 is also interacting with the specific receptors. Those are the ryanodine receptors of this smooth endoplasmic reticulum. These ryanodine receptors will cause the release of calcium from the storehouses into the cytoplasm, and when this calcium will move into the cytoplasm, it will do its function via calmodulin and calmodulin dependent kinases, so CAM kinase. So this is a crosstalk between the calcium signaling and the PLC beta 1 signaling. And we also know that calmodulin dependent kinases are able to affect the DNA expressivity and its transcriptional and translation things via CREBP. So this is the point of conversion here as well. So we can see that crosstalk between calcium signaling and PLC beta 1 signaling and the convergence into the CREBP regulation of the function. The whole sequence of calcium, which regulates a number of different kinds of receptors and number of different kinds of signalings, can be seen in this picture, where calcium regulates the expression and the response to different kinds of neurotransmitters as well as certain antigens as well. So it's a varied kind of an signaling which combines a number of different kinds of responses in the target cell. So this was pretty much it about the cGMP and calcium signaling. We'll be covering receptor tyrosine kinase signaling in the next lecture to come. Do visit my Amazon page for the various books which are available for the biochemistry and molecular biology subjects. And do subscribe to my channel for further videos which I will upload in due time.